Buongiorno, buonasera, buonanotte, buon pomeriggio, a seconda del fuso orario da cui state guardando. Questo è un altro episodio di Liberi Oltre STEM. Questa volta siamo con un gradito ospite, il professor Sean Booth, dall'Università di Manitoba, Canada, che ha pubblicato recentemente un interessante articolo sull'evoluzione degli arsenali microbici nei patogeni, specificatamente Pseudomonas aeruginosa. Questo articolo è apparso su Nature, uno dei giornali di Nature. È molto interessante perché unisce una parte di simulazione a una parte di modello, di esperimenti. E Sean è il primo autore di questo articolo, quindi abbiamo invitato per una breve conversazione, che sarà ovviamente in inglese, che comunque potrete sottotitolare con YouTube, per raccontarci come funziona il suo lavoro, quali sono gli strumenti che utilizza regolarmente, come è nata l'idea di questo articolo, come l'ha sviluppata, quali sono gli strumenti di controllo e come in generale si può impattare in una ricerca così competitiva come quella di biologia un'idea nuova e che può portare a degli sviluppi interessanti. La sua ricerca di base quindi non è immediatamente applicabile, però è sicuramente molto interessante. That said, here is the floor is yours, Sean. Please start maybe introducing yourself for our public.
Yeah, so my name is Sean Booth. I'm now an assistant professor at the University of Manitoba in the Department of Microbiology, and I'm actually just moving there. I am moving from the University of Oxford, where I was formerly a postdoc in the lab of Dr. Kevin Foster. And yeah, so I'm quite interested in how bacteria interact with each other, and so like other ecosystems where we can see animals like in the savannah where you have lions eating zebras and the zebras are eating grass, bacteria also interact in similar ways where they come into competition with each other. And so I'm really interested in understanding how bacteria engage in these very aggressive interactions in order to try to really take over an environment and maximally exploit resources.
You mentioned predator and prey, but it's not just predator and prey relationship in bacteria. There is more complicated interaction.
Right. Yeah. So it's more like a peer-to-peer interaction. So these bacteria aren't eating each other. I mean, predation does exist in bacteria, but really it's more competition for the same resources. So you might, to continue with the analogy, say the competition between like hyenas and lions, or maybe between different animals that are eating the same plants. So these bacteria, they're in the same environment and there's some limited resources. And so they're competing for these resources. And one of the best ways to exploit those resources is to just eliminate the competitor. And so to do so, bacteria have evolved a wide arsenal of different weapons. And so these weapons are molecular systems that have specifically evolved in order to kill competitors. So their function is to kill competitors, not to eat them, just to take them out of the picture, so that the producing bacteria can then have the territory to themselves.
May I ask you something? We often see other strategy of, let's say, of contrast to the bacteria. For example, finding a different ecological niche. The plasticity of bacteria make them adaptable in a complex stratified environment like subsurface environment to find a niche in which the other competitor cannot enter.
Right. Yeah. So, for example, using a different electron receptor. So a lot of bacteria use oxygen, but there's other possibilities. So say using nitrate or respiring different kinds of compounds. So in the classical, what are they called? The columns.
Winogradsky column.
Winogradsky, yeah, exactly. Where you have these different colored bacteria and they're using different electron acceptors. And so now instead of competing with each other, now they've now stratified and they don't have to come into competition. And the same thing can happen with different carbon sources. So if some bacteria eating some sugar and the other bacteria eating some amino acid, then they don't need to come into competition. So they often use motility, chemotactic motility, so they can sense these compounds in the environment and move towards it. And so now just by using that ability to move towards the compound, they're moving towards their favorable niche and maybe can then escape the competition that might be happening somewhere else.
But unfortunately, sometimes war is inevitable, even within bacteria. So when they are in the same environment, for example, the host, because we're talking also about pathogen, or in niche when it's not possible to find other electron acceptor, other carbon source, then they have to compete, and they compete through a range of mechanism that you briefly introduce in your paper, like bacteriocin, talocin, so bacteriocin tailored to specific species, long range, short range. When it comes to long range, short range, you come with a very subjective classification. Maybe you can tell us more about that.
Right. So this paper, we originally put it on the bioRxiv preprint server. We called it Bows and Swords, and so we're referring to how bacteria have weapons with different ranges. And so, like a sword, it requires you got to get up close and personal. And so bacteria, similarly, they have weapons that require cells to be right adjacent so they can stab their weapon into it. But they also produce, as Enrico mentioned, these different weapons, bacteriocins and talocins. So these are released into the environment and they can diffuse at a far distance away from the producing cell, and so then affect distant cells. And so that's quite a contrast, right? And that's kind of where we came into this question was, well, okay, very close, very far. Why do we have bacteria that are producing both of these different weapons?
Okay, you say in the introduction that why bacteria produce such a range of weapons remain a mystery. Now, maybe I misunderstood this statement, or maybe that was kind of a bit too strong. It is not entirely a mystery why, the big reason. Maybe the details are a mystery in a specific environment, but of course the reason is defense. Am I correct?
Well, that was kind of a question we were wondering about, that there's kind of been a thought in the field that like maybe some weapons are for defense and maybe some are offensive. We weren't really sure that was the way to be thinking about these bacterial weapons. So one example of this is the type 6 secretion system. So some bacteria, this is a close range weapon. It's kind of like a spear gun where they fire this needle into an adjacent cell to inject a toxic protein. And so some bacteria, they just fire randomly, hitting anybody nearby. But some Pseudomonas aeruginosa, they only fire their weapon in response to an incoming attack. So you could kind of think, oh, okay, this is a retaliation. But that's just within this one category of this close range weapon. It's a very specific situation. Whereas, this other way to classify different weapons based on their range, and we know bacteria have different kinds of close range weapons, and they have different kinds of long range diffusible weapons. And so rather than thinking like, okay, are they producing this to defend from an incoming attack, or are they producing this to just randomly attack whoever's nearby? Let's frame this in a different way. Why are we doing things that act close and far? And why do we have a single type of bacteria that produces both of these? So they have both, which means there's some reason to have both.
Okay, so basically the idea of the paper comes from a reasoning of theoretical biology and theoretical evolutionary biology, and then you come with a slightly new classification. Rather than classifying by chemical structure or by effect, to say defensive, offensive, you classify out of range, which is actually an engineering classification, if you like, physical classification, because diffusion, molecular diffusion versus contact, which is actually not mediated by diffusion, is instantaneous in a way. So instantaneous effect versus delayed effect. And then you find, you try to approach from a modeling side. So how you come out with the idea of using agent-based modeling?
Yeah, okay. So as I mentioned, we have this one bacterium, Pseudomonas aeruginosa, and we know quite a bit about it. It produces both short-range and long-range weapons. And we also know a lot of bacteria produce various different long-range and various different short-range weapons. But each of these different weapons, they're quite different from each other. So there's a very wide spectrum of how they work exactly. So, for example, the diffusible weapons, you have things like antibiotics, which are small molecules. So like streptomycin is like quite a small molecule. Then you also have bacteriocins, which are protein toxins. So it's just like a normal ribosomal protein released by the cells. It still diffuses, but it's much larger. And then you have even larger are the talocins. And so these are actually bacteriophage, so viruses that infect bacteria. that have been kind of domesticated and repurposed as a weapon for the bacteria. And so all of these act at a distance, but we know that they're going to have very different properties. And this is kind of the same case for various different close range weapons. And so it's very hard to go out and say, okay, how can we perform some experiments to compare this huge range of diversity? But with the model, so the way the model works, it's an agent-based model, as you mentioned. We have individual bacteria, and they're each computationally simulated. So they grow and they divide and they behave like real bacteria, only they're in a computer. And then then we program them to say, okay, you can produce a long-range weapon or you can produce a short-range weapon. And what this allows us to do is to make them as similar as possible. So both the long-range and the short-range are now going to be equally effective, and they're going to be produced at exactly the same rate. And this is something that, essentially you can't do experimentally. And it allows us to now ask a much more general question to say, okay, just the only difference is the range. Everything else is identical. And we can only do that by, simulating it computationally. And this allows us to kind of get an idea of what's happening with the biology.
Let me tell you something. I did a bit of work a long time ago with the ABM using a simple language called NetLogo, which is basically a toy. But one of the things that you notice that the model is as good as the parameters you put in. So what you are saying is, in my opinion, very important. The only difference was the range. So every other confounding factor was removed from the model, and you can just focus on the effect of short range versus long range in terms of emergent properties of this biofilm, so these material aggregates. Because if you introduce other model, the other parameter, the model becomes so variable that you can basically force and torture your parameter in order to obtain any conclusion you want. Instead, by using it, you give a certain rigidity to the model so that the response is probably more close to reality. That's my understanding, I think.
Yeah. So you really want to try to limit the things that you can change, because I mean, the model you can also run much more quickly than an experiment. It's run on a computer. Running a lot of different conditions takes time, but just one condition is very quick. But once you change a few more parameters, it starts to balloon and balloon once you start to change many different parameters. So if you just limit it to like what we were interested in was this range thing, then we could just check how that affects the outcome of the competition, and everything else is kept the same. And so it also, it kind of contains the scope of the simulations.
Okay. And then you come into a variety of illustration, very colorful picture. To be honest, very easy to understand, because the work that you have done before is massive. So the final results is actually understandable, even by not say a layman, but a person that has a undergraduate education in biology is quite clear. And you come with some very interesting results. So what are the main finding of the modeling part?
Right. So the main finding from the model was essentially that the short range weapon. So what one of the parameters we were interested in was, okay, if we have two bacteria together, one which has the weapon and one which is susceptible to the weapon, it's sensitive. So the attacker can kill the sensitive strain. And we wanted to know, okay, if we change how much of each strain there is, so we can say there's lots of attacker or there's not very much, how does each weapon perform? And what we found was that the short range weapon, under all conditions, it worked fine. So even when it was outnumbered, it worked great. But the long range weapon, it did not. That strain had to have a numerical advantage. And the reason for that is that if you have a lot of bacteria that are producing this toxic substance, it can accumulate in the environment and it can exert its effect. But when there's only a few of these cells, it just diffuses and it never reaches a useful level. But the short range weapon, if they're outnumbered, it doesn't matter. There's going to be somebody nearby and it's going to work. And so that was quite interesting that we now see this big difference in the frequency of the attacker strain, how each weapon is useful.
So to put it in a more human perspective, it's like in-course strategy, Navy SEAL strategy versus a large army that invade the whole country. A large army invader a country, you need to be big, need to have a lot of resources, because producing a toxin means that in your model, at least, means that you grow less if you produce toxin. You're playing like a role game, in which you have less point for production and more point for life, and vice versa, which is super fascinating, actually. So short-term, shorter range strategy always work, but it has also short-term effect on the short time. So you need more time to exert an effect. While long-term, it's work only when you have many bacteria, because only in presence of many bacteria, the concentration of your toxin become high enough to kill the susceptible cells, right? So in the end, it look like the best strategy appear to be the short contact, at least for the beginning of the invasion of the host, or the beginning of the invasion of the place where the other microbes were living in peace. Okay, that's super cool, actually. I think that I have here a slide. This is one of your figures, so show the short range. And here you have in tracer simulation, sometimes the stabbing work, sometimes the stabbing does not work. So you account for that sort of success rate of stabbing. And of course, the diffusible toxin is produced at anytime, regardless of the result of the attack, of the outcome. So there's a continuous cost, while the lethal toxin costs only when it's produced and when it's stabbed. So it's slightly different parameter, right?
So what we did was the secretion rates were matched for both. So what this means is that for the short range weapon, they're releasing, we call them firing events, and so as you said, they can miss, whereas the diffusible toxin is released but accumulates in the environment. And so this is what the simulation is really great for, is it allows us to match it exactly the same. So both toxins are being produced at the same rate. And as you mentioned, we have this cost, which is shown on the right side, that the more toxin being produced, the less the cells grow. And this makes sense, right? If the cell has finite resources and it's investing more in producing the weapon, it's going to be able to have less resources available to invest in growth.
Our channel was born, the association was born from a group of economy professor. So this is called also cost opportunity. Either you spend in producing weapons or spending improving your life. You cannot do both. And you have to manage the frequency of doing these two actions. And that's another result. I'm just going through the figure of your paper. And we should show at the snapshot, so at the end of the simulation time, I don't know how much it was, what is the situation with short-range weapon on the left and the long-range weapon on the right, for the same population of attacker/invaders, right?
Yeah. So these simulated biofilms, they, regardless of whether it's a short-range or long-range attacker, they start from the same condition, and then we allow them to grow up and engage in this competition. And we can, it makes it very easy to see what happens. We can quantify it as well, but just looking at the outcome, you can see very quickly that at like the equal frequency there, everything's blue, which means the attacker has killed off all of the yellow sensitives. Whereas with the diffusible weapon, they're kind of still equally matched at 1-to-1, even though that attacker has had time to attack.
Yeah, and of course, what is called competitive advantage is a parameter, variable or parameter that you arrange to show the maybe how many more units of the attacker are born or surviving. I don't know.
Yeah, so I guess the exact technical definition is that we know how many cells we have to start, and we know what the ratio is. So, for example, in the middle there we have 1-to-1. So even if that's 100 of each, it's still a 1-to-1 ratio. And then at the end of the simulation, we can count the number of each kind of cells. And then what we do is we take that final ratio divided by the initial ratio, and now we have kind of fold change. And so on that competitive advantage is on a log scale. And so what that means is that on the right-hand side there, where the contact attacker is winning by a lot, it's winning by far more. So it's a 10,000-fold advantage compared to, the 100-fold advantage you see when we have a low initial density.
Okay. And the attacker frequency is the rate at which the chemical attack, either diffusible or contact based, is carried out. So it's basically the frequency of the aggressivity of the attacker on the x-axis.
No, so the
sorry.
attacker frequency. This is like in the population. So population frequency means how much of the population is one strain. So 50% attacker frequency is equal. So that's 1-to-1. So it's 50% attacker, 50% sensitive. And then on the left, we have 10% attacker, 90% sensitive, and then 90% attacker, 10% sensitive. So as we go from left to right, we're increasing the initial amount of attackers. So you can see, I guess, kind of, on the left side there of the diffusible, you can see that there's not very much of the pink because there was only 10% to start. And then in the middle, it's about 1-to-1. And then at the bottom, it's 100%, but it started from 90%.
So the initial attackers susceptible on the left part of the figure is the same as initial attacker frequency on the right?
Yeah, exactly.
Okay, that was a bit misleading, sorry. So what is initial density? Just the number?
Yeah, so you could say if you have 100 and 100 or 10 to 10, you still have the same ratio, but the density is different. So in the one case, there's 20. In the one case, there's 200.
And technically, if I understand correctly, the density shouldn't affect the short range, but should affect a lot the diffusible, because it exactly increase the diffusion, decrease the delay the diffusion, correct?
Yeah, and that's what we found, is that the diffusible weapon needs the high density in order to be effective. Okay,
thank you. And then this is instead the time evolution. So you did first the snapshot at the end of the simulation, and then you show the situation in dynamic by time, I don't know, 0 to 5, this is hour, yes. And show what's happened as the time goes. Maybe you comment on that briefly?
Right. So in this experiment, we wanted to know what happens if we start from an already full population. So you mentioned kind of an invasion situation. That's what we were wondering about. So if we already have the susceptible strain has already occupied this environment, can the attacker invade? And so we see at time 0 here, we've added the invader in, and then we added that at a relatively low frequency. So this is, I think, 10%. And then we can see, okay, if we added at 10% frequency, when it's already an established biofilm, what happens? And so we see with the contact weapon, you get this increase in the amount of blue until eventually it's taken over the entire population, whereas the diffusible attacker is not able to invade. And this is the case where we're trying to look at the different production rate of the toxin. So on the right side there, the secretion rate, this is the rate at which the toxin is being produced. Because we weren't sure. It's like, okay, maybe you only need a little bit, maybe you need a lot. And we tried to change this equally for both short and long range weapons. And what we can see is that the contact weapon doesn't matter. No matter what secretion rate, it always works. And whereas we get the opposite for the diffusible weapon. No matter how much you're investing in producing this weapon, it never works because you don't have the numbers to make use of it.
So first you need to multiply as an invasion strategy and then to use diffusible. So there is also a time lapse between the two. This is fantastic. This is quite kind of counterintuitive. I mean, unless you do the number. So it's for me, one of the fun results of this modeling part. And yes, you have just a slight advantage, a very low production rate of toxin, but as soon as you produce more, you spend more and you lose your competitive advantage. Very nice. But then eventually you start doing experiment, which raises one question. Where do you get the data, the parameter for the modeling? From the experiment or from the literature?
So the modeling was done. Yeah, this is important. The model was done before the experiment. So we wanted to say, like, without knowing what was going to happen, we can use the model to say, okay, what's happening. And so we're not. So the growth of the cells and the division rate and the size of the cells, that was taken from literature values. But so it's not matched up to what we did here in the experiments. The experiments are a kind of a specific test to what the model is a general case.
Okay. So what the experiment show in, there are 2 or 3 slides for the experiment. This is the, as you remember, this is the 2 strategies when you have susceptible cells, false color in yellow, and the 2 attacker which are separated. So basically you have from Sujata, by the way, I think one part only with one weapon, another part of the separation with another weapon only. So you're able actually to split genetically the 2 strategy in the same strain.
Yeah, exactly. So I genetically engineered separate strains. So on the top, there's no yellow cells. You can't see them because they've been killed off, but they were genetically engineered to be susceptible to contact-dependent inhibition. So this is the short-range weapon that we used. It's different than the Type 6 secretion weapon, but secretion system, but it's a similar weapon. And then on the bottom there, we can see the yellow. And in that case, it's actually a different susceptible strain because the sensitivity to the taylosin works in a different way than the sensitivity to the contact-dependent inhibition. So yeah, once I genetically engineered these strains to make them susceptible to the weapon, I also added these fluorescent protein genes so we can tell them apart. So one was yellow, one was red, but with the microscope data, we can make them look whatever color we want.
Yeah.
We have nice blue attackers with CDI and our purple, pink taylosin attacker. And yeah, so what we find is as we increase the density, like we just talked about, so in both cases here, the ratio of attacker to sensitive is the same, but we increase the density, and we can see more clearly with the taylosin attacker that at low density, there's plenty of yellow cells. So the sensitive strain is doing fine, but we go up in density and we start to lose this yellow color, which means that the purple attacker has killed it off.
But then something start happening, which probably prompt the next set of experiment. You start to observe a spatial distribution in the colony. These are all colonies on a plate, agar plate, and you start to observe a difference between the center of the colony and the edge of the colony, which is due to diffusion and to the way in which bacteria grow. Because bacteria grow from the center of the colony, expanding to the edge. I guess this prompt the next set of experiment, which is this. In which you see you determine the competitive advantage and in two parts of the colony. Is that correct?
Yeah. So like you said, because the colony is growing outwards, and plus we could see it in the microscopy images that the center of the colony is quite different from the edge. And so what I did was I used a little pipette tip to take some of that colony material from the middle and some from the edge, and then I counted the number of cells that were yellow and red, one of representing each strain. And then I did the same math we talked about for the simulations to see the change in ratio from the beginning to the end. And that gives this competitive advantage. And what we can see is that at the edge, the contact-dependent inhibition is much more useful compared to the diffusible taylosin. So we can see that kind of matching the model that even at low attacker frequency, the contact weapon still being super useful, especially at the edge of the colony.
This goes a bit along the work of people like, not only him, but like Stefan Wurz with the concept of rural and colonizer, if I remember correctly. So in a given population, the edge and the center, they work also showing the faster bacteria at the edge of the colony. You remember this work done at UCLA. Basically, the bacteria in the center are stationary. They stay cool. They just grow, while the bacteria at the edge are the incursor. So they go around, kill, and find resources due to physicochemical reason. So you reproduce this kind of situation, apparently.
I'm not familiar with the work you're talking about, but
okay.
There's definitely this difference between the center and the edge.
Basically, the one outside the colony are faster in this work that I'm citing, faster, meaner, and use more contact weapon because their job is to expand to the next environment, while the one inside build the colony, build the biofilm, by the way, and are more focused on producing the matrix than expanding. I'll send you some paper later. It's actually fascinating, and I think it intersects very well with your work. Anyway, sorry for this. The last thing is then, of course, you put them head to head and see what happened. It was fascinating.
Yeah, so I mean, this experiment, I guess we hadn't really thought of this question until we started the study, but it was like, okay, well, now I have the ability to make these strains. I can make one strain that can kill the other with each weapon. So one has one weapon, one has the other, and it's like, let's go, let's see who wins. And so yeah, that was just a really fun thing to do. So actually, in this case, I made the strains, and then it was like, okay, we need to model this because we hadn't done it yet. Because I'm going to do the experiment, so we need to also have the model to see what happens. And yeah, we kind of saw the same thing again, but the cool thing here was that initially it seemed like, well, why do you have this diffusible weapon? Because it seemed like the contact weapon was just always better.
Yeah.
But what we found here was that actually the contact weapon, you can see it on the bottom left there on the left side, that even when it starts at a high frequency, it's not doing much better than at equal frequency. Whereas on the right, the Taliesin, where it's way down at the bottom there in purple, there you can say, okay, yeah, now the Taliesin is doing really well. So apparently the diffusible weapon does really well against an armed attacker, whereas the contact weapon, if the strain is fighting is armed with diffusible weapons, it's not very useful anymore. And so this kind of showed, okay, look, now we have a good reason to have each weapon because, this diffusible weapon, very good fighting against an incoming attacker.
Yeah. And then we go, of course, to the final conclusion. The right say 2 weapons is better than 1, which echo a famous commercial in Italy, but it makes sense. So once you have two strategies, in general, you win. And that's exactly what happened in this bacteria.
Yeah, exactly. I mean, yeah, this was another case. It's that, okay, we have the ability to do this experiment. Let's just make one strain that's got both, one strain that's going to just get pummeled and see what happens. And we saw it exactly as expected. 2 is better than 1.
Yeah, of course, this is a one specific case of one pathogen, but the implications are serious because is a potential approach, a potential strategy for many organisms, not just pathogen, not just this Pseudomonas aeruginosa, but other bacteria, biofilm producing, non-biofilm producing, and can be interesting maybe later on, maybe some sort of a meta-analysis, repeating this for 10 different species and see if there is any trend that emerge. Of course, maybe your future work, I don't know, but it's a massive work. And we found this paper interesting because it's just unusual. My personal comment would be super cool to see what happened in 2 different metabolic niche. So how this competition shape at the border between oxygen respiring and, I don't know, a nitrate respiring bacteria, like in wastewater. But that's a completely different work. So it's just a curiosity. So yeah, what do you think of the future relevance of this work, in your opinion?
Yeah, I mean, this was kind of the first foray into trying to understand this kind of broad question of why do bacteria have so many weapons? And so we took this initial framing of, okay, why short range versus long range? But I didn't really mention this, but bacteria also carry multiple versions of the same weapon. So you have bacteria that have several short range weapons at the same time, and they have several long range weapons that are actually, very genetically similar. And so the question is, okay, well, now we've found this explanation for why you have short versus long, but why do you need multiple similar ones? They look like they're the same. Not really sure why that's the case, and so that's a question I'm looking into now to see how this happens. And we talked a lot about the center of the colony versus the edge, right? And you mentioned motility. So bacteria like Pseudomonas aeruginosa, they have a kind of miniature grappling hook that they send out, and they can pull themselves along to crawl along the surface. And what we wanted to know was, okay, well, if bacteria are moving around, but they're also fighting each other with cells that are nearby, this ability to move is probably going to be important here. And so that's actually a study that is pre-printed and it's under review right now, where we tried to ask this question, okay, does motility affect short-range weapons?
Okay. So basically, another aspect I find interesting, and then we're going to the conclusion, is that it's not just bacteria on an agar plate, it's not just bacteria on liquid. the interaction with bacteria can now be visualized in 3D with the cell precision, so you can actually track thousands of cells at the same time, and you can track also biochemical production of arsenal in the time using single cell transcriptomics and so on. So we are going to have a very precise understanding of how aggregate work. One of the sentence of the paper said that in contrast with what we know before, bacteria are not individual but are groups, and so they tend to have group strategy. And your work fit there, indicating that some of the attacker will die, but the species will procreate, which is of course Darwin's initial intuition. But also this refers to the biofilm and how bacteria make a house for themselves, and how this fighting come with the building their community. So, which is another interesting aspect for evolution.
Yeah, I mean, one really interesting connection there is that so these cells that are producing the telogens, they actually have to burst to release them. So that cell is now dead. But
okay.
its siblings can then benefit from the production of the telogens. But also, when it bursts to release the telogens, it also releases its own DNA, which is like physical structural component of biofilms. And so now we have this one behavior that's kind of doing two things at once in the biofilm. It's attacking other cells, but it's building the physical structure too.
Okay, the level of complexity of this thing is spiraling, sometimes out of control, and that's why we need good interaction between computer science, because we can do experiments faster and many variables, but good confirmation with the proper genetic engineering and so on. So basically, the modeling shed light where you want to go with a single experiment. You can do 100 experiments, and then the one is most interesting, or the 2 or 3, you do it in real life, which is one of the direction. Okay. It was a fantastic talk. Thank you very much, Sean. Let me summarize. Professor Sean Booth from the University of Manitoba. And now, if you want just to, if you allow me to share your email or a QR code somewhere that people can actually contact you. I suppose that you are recruiting people from your group right now.
Yes, I am. I am recruiting grad students for, I guess, the summer and fall 2024.
Okay, and if you want to work with a very good scientist and also a very good person, please email Sean and see if there is a spot for you. It's a very, Canada is a very nice place to live, I should say.
Yeah.
Okay.
It's a bit cold in the winter.
It's okay. I mean, it's cold. It just means more heating, and there's a lot of tunnels in Canadian city to go underground when it's very cold, so it shouldn't be a problem. Okay. With that, I think we can conclude. Thank you very much, Sean, for your time. Congratulations for your paper, and I will check now your preprint and maybe send a couple of papers that we talked before. And so long. See you next time.
Great. Thanks a lot, Enrico. It's been great talking to you.
Buonasera a tutti. Arrivederci.